And this is my best friend, Boots. Hola! Do you like exploring? Yay! Great!
Dora! Dora!
Hey, Boots, did you hear that? Yes! I did too!
Dora! I need your help!
Who are you?
I'm Ari Patronos, and I need your help, Dora!
Why? What's the problem?
I lost my map!
Could you help us find this map? Great! What does your map look like?
It's a human genome map!
What's that? Don't you know, Dora? It's the one used in the Human Genome Project.
That's right, Boots! And our goal is to identify all the 20,000 to 25,000 genes in the human DNA to determine the sequences of the 3 billion chemical base pairs that make up the human DNA, and to store this information in that database. And I lost that database!
But how will we know where to go? Who do we ask for help when we don't know which way to go? The map! Right! Say map! Say map! Louder! Louder!
I'm the map, I'm the map, I'm the map, I'm the map, I'm the map!
Hey map! We're looking for Ari's lost map!
A lost map? That's bad. Okay. According to my GPS, you have to go through the DNA Creek, then pass through the Controversy Mountain, then dance your way through the mapping road, and you'll find the map 3 steps to the left at the top of the thing in the corner of that place right there.
Come on, Baba! Everybody, let's go! Come on, let's get to it! I know that we can do it! Where are we going? To the bridge! Where are we going? To Ari! We left him! Come on, let's get to it! I know that we can do it! Where are we going? To the bridge! We made it! Look! There are missing steps in the bridge!
It's not a bridge, it's a Leopleurodon!
A Leopleurodon!
Yes, and the missing steps are its genes. We have to complete it.
Will you help us match the nucleotide bases? No? Great!
You know that we are all made out of cells, and each cell contains a genome, a complete set of hereditary information called genes of that individual. These genes are made out of repeated sequences of 4 chemical bases, namely thymine, guanine, adenine, and cytosine. Genes carry information for making all the proteins required by organisms. These proteins determine, among other things, how the organism looks, how its body metabolizes food, and etc. It can even explain how it behaves sometimes.
Does that explain why you're ugly, Mr. Patronos? Dora, don't say that in front of him! Could you please turn around, Mr. Patronos? Does that explain why you're ugly?
Cut it out! Did you know that this DNA could also be used to serve as a fingerprint to determine the relationship between 2 organisms? Even you and me? Even you?
The genome. If we could journey inside ourselves into a cell, we would see 23 pairs of chromosomes packed into a nucleus. Each chromosome contains a long coil of DNA. If all the chromosomes were unwound, the DNA in just one of our cells would stretch 6 feet long. The DNA double helix contains 4 kinds of building blocks. An A always pairs with a T, a C with a G. DNA contains information to make every part of our bodies with its 4-letter language. Each of our thousands of genes codes for a specific part.
Yay! We did it!
Do you know where
Do
we're
you
going?
know where we're going next? Alright! Let's go! Look everyone! There's a troll by the door!
Oh no, a troll!
What is the password?
What password?
I guess it's the goals of the Human Genome Project.
What made you think of that?
So the other goals are to improve tools for data analysis, to transfer related technologies to the private sector, to address the ethical, legal, and social issues that may arise from the project. Also, with the Human Genome Project, molecular medicine can improve the diagnosis of the disease. It can also detect diseases earlier. Radiation drug design, controlled gene therapy, and even cure genetic and acquired diseases. Knowledge about the effects of DNA variation among individuals can lead to a revolutionary way to diagnose, to treat, and someday prevent thousands of disorders that affect us.
Can you please let us in, Mr. Troll?
No, I want password.
Can you please tell us what the password is?
Correct. You may go in now.
Now we can pass through Controversy Mountain! Oh my, holy cow! There's a lot of controversies and issues. We have to solve them to get through!
To learn all about these controversies, watch this video.
The scientists who launched the Human Genome Project believed in the power of genetic information to transform healthcare, to allow earlier diagnosis of diseases than ever before possible, and to fuel the creation of powerful new medicines. But it was also clear that genetic information could potentially be used in ways that are hurtful or unfair. For example, denying health insurance because of an increased risk for developing a particular disease. Aware of the danger and hoping to ward it off, the founders of the Human Genome Project created a program to explore the ethical, legal, and social implications of new genetic knowledge. The goal was to anticipate problems that might arise and to prompt solutions. For example, in the future, doctors will likely be able to give each of us a genetic report card that will spell out our risk of developing a variety of different diseases. But will we really want that information? How will it be used? Who will have access to our genetic information? How will it affect our lives, our families, and our communities? The challenge of addressing these issues is not reserved for scientists. We all have a stake in making sure that everyone will benefit from genetic research and no one is harmed.
Let's go!
Swiper! What do you want? We don't have anything! Ah! Come on! Swiper, no swiping! Swiper, no swiping! Swiper, no swiping! Oh man!
Alone? Dora? Boots? Okay, I walk alone. But first, I have to explain the project and how we began. So, what we did was we made maps of the human genome. First, we had to identify DNA sequence markers to help us navigate across the chromosomes. Don't forget, these markers should be polymorphic You don't know what it means, do you? It means that alternative forms must exist among individuals so that they are detectable among different members in the family studies. First, we use gene linkage maps. We created these maps based on the recombination frequency, which is basically the frequency that a single chromosomal crossover will take place between two genes during meiosis. When double recombination occurs, it would seem therefore that no recombination occurred. When the distances become higher, the likelihood of a double crossover increases. If the loci we're analyzing are very close, double crossover is very unlikely. So therefore, genetic linkage maps are constructed by observing the first two markers that are inherited together. It's easy, isn't it? No, just read the handout.
Yes, also, physical maps vary in three major types based on resolution. Chromosomal or cytogenic, which is based on the distinctive banding patterns observed by live microscopy of stained chromosomes. Radiation hybrid maps, where scientists use breaks induced by radiation to determine the distance between two markers. Sequence tag site or STS mapping, where overlapping DNA fragments are cut using restriction enzymes and are noted which fragments contain STS, a short unique DNA sequence of which the exact location and order of the bases of the sequence must be known, cloned using recombinant DNA technology. These unordered cloned DNA fragments are called libraries, then are introduced to a suitable host cell to reproduce and are assembled in the order they would be found in the original chromosome by determining which clones contain overlapping DNA fragments, the assembly of which is called a clone contig. Scientists also use a technique called chromosome walking, where everything around a known piece of DNA, the starting probe, is cloned. All clones hybridizing with the probe are screened in a genomic library. The most distal piece of this most distal clone is then used as a probe, so that ever more distal regions can be cloned.
Success! At last! Now we just have to find the map. 3 steps to the left of the top of that thing, next to the corner there.
Wow! Great memory! Yay! We did it!
Sing along, everyone! We did it! We did it! We did it! Yay! Ellie, we did it! We climbed the tallest mountain and swung from tree to tree. We did it! We did it! We did it! Hooray! Outer space into the bottom of the sea. We did it! We did it! We did it! We did it! We'll always stick together, run, sing and play. I'm so happy to you a happy birthday.
What was your favorite part of the trip? I like that too. My favorite part was when the corporate child got hit by a car.
That's not part of the... Seriously, that's not part of it, is it?
Smart, you know with me and you and our friend Blue, we can do anything that we wanna do. We can do anything that we wanna do. Bye-bye. Thanks for all your help.